the bacteria gastrointestinal infection is the microbial disease of the digestive system, whereby inflammation occurred in the stomach and intestine of a host. When the bacteria is in the gastrointestinal tract, its main goal is survival. Therefore, each species of bacteria has their own way of exploiting and manipulating the cellular function of the host for their benefit. An important element that occurs in almost all bacterial gastroenteritis problem is that these bacteria aims to control the water regulation in the host gastrointestinal tract, as pictured on the right. By manipulating the environment, the bacteria benefit by being able to outcompete the microbiota of the host, and is then able to disseminate into external environment. On the other hand, the host will also use the water regulation in the GI tract for the expulsion of the bacteria or any unwanted organism out of the system. With this synergy, diarrhea is generated, which makes sense, as diarrhea is the most common response of gastrointestinal infections. Other symptoms present would be abdominal cramps, nausea, vomiting and fever. As a medical officer or a public health officer, the understanding of how the bacteria transmit from one place to another, its pathogenesis, and the prevention method is crucial to stop a potential outbreak. Most of the common bacteria involved in gastroenteritis, like the Salmonella, E. coli and Vibrio cholera, can be isolated from the environment, or from the infected person themselves, and be identified using conventional biochemical tests or PCR, depending on the laboratory's budget. PCR would produce a more concrete confirmation, as their genetic information is obtained. Other than that, the post-field gel electrophoresis or PFGE can be used to determine the genetic relatedness of the bacterial isolates. This is helpful to determine if one outbreak is related to another outbreak, or where is the source of the bacteria from. A disk diffusion method can be used to know the level of antibiotic resistance of the organism. This is important, as there are cases of the same strain of bacterial outbreak that occur at different years, but the same bacteria has since evolved to develop characteristics of antibiotic resistance. One of the most discussed bacterial gastrointestinal infection is the cholera disease, caused by the bacteria Vibrio cholera. According to the World Health Organization, cholera differs from other acute diarrhea, in that it occurs in large epidemics that involve both children and adults. The infection of Vibrio cholera would cause a profound watery diarrhea that could lead to extreme dehydration with hypovolemic shock, which can be fatal. The diarrhea has the characteristics of a rice water, as in there is no fecal particles, is watery, and is flecked with mucus. One of the characteristics of Vibrio cholera that should be noted is that they survive in fresh water for a period of time and in salt water for at least a week. Which makes sense, as most cholera cases come from the consumption of undercooked seafood. Even though they are resilient in their survival, this organism could also be easily killed by heating at high temperature, through chemical actions, or by introducing a competing bacteria. As was previously discussed, Vibrio cholera can be isolated from the diarrheal discharge or through a rectal swab of a suspected patient, and be identified using conventional biochemical tests. To get a more solid confirmation, the PCR or PFGE can be used. One of the main incidents of cholera in Malaysia was an outbreak that happened in Terengganu in the year 2009. The cholera was spread to several districts within a week. Approximately 400 persons were hospitalized for treatment of acute diarrhea and its complication during the outbreak period, with a record of 187 confirmed cases and 1 death. It was discovered that the main vehicles of transmission in this case was the contaminated drinking water, and the consumption of raw or undercooked seafood. One of the causative agents of salmonellosis, that is most commonly reported as Salmonella typhi. It is a toidal salmonella, that causes typhoid and paratyphoid fever. Salmonella typhi is only restricted to growth in human hosts, and its principal habitat is in the gastrointestinal tract and the bloodstream. The symptoms show up about 1 or 2 weeks after ingestion of suspected contaminants. The symptoms ranges from having headaches to fever, anorexia and mental confusion, to having visible rose spots on the skin and diarrhea. Most cases of salmonellosis results from the digestion of contaminated food, especially the poultry and inadequately pasteurized milk, Typhoid fever is still a concern in developing countries with a World Health Organization record of about 16 million clinical cases and 600,000 deaths. This stems from the lack of antibiotic supply, proper water filtration system, and sanitation. The diagnosis of Salmonella typhi can be done from getting stool or blood specimens, and be tested using the same method as previously discussed, such as the biochemical methods, PCR and PFGE. In the year 2005, the State Health Department of Kelantan reported 517 confirmed typhoid cases with 2 deaths. It was said that the outbreak coincided with a festival. The serotype of the isolated Salmonella typhi was reportedly the same strain as previously isolated in the year 2003 and 2004. The source of the bacteria came from food samples, drinking water, and the food handlers, but the key point here is the poor sanitary condition, lack of treated quality water, and the involvement of the food handlers as carriers. With that, some of the actions taken by the Ministry of Health Malaysia was vaccinating the food handlers, giving health education and public awareness on personal hygiene, and closing down a number of food premises. The bacteria E. coli is most often talked about among causes of gastrointestinal infection. One of the E. coli serotypes, called the E. coli 0157H7, is often implicated in illness worldwide as sourced from the Center of Disease Control. It has features of enterohemorrhagic, and it produces a toxin called Shiga toxin that damages the gastrointestinal mesochal tract and vessels, and other organs, especially the kidney. This organism has a cow reservoir, which explains why it is usually found in unpasteurized milk and undercooked beef. It causes a big concern because, as low as 10 cells of E. coli 0157H7 is enough to cause an infection. According to the guidelines from the United States Department of Agriculture, a ground beef is considered adulterated, or to be discarded from human consumption, if as little as 1 colony-forming unit of E. coli 0157H7 was detected in 25 grams of ground beef. Upon infection, the symptoms shown are severe abdominal cramps and watery diarrhea, that progresses to bloody diarrhea. The infected person would also have low-grade fever, that lasts for approximately a week. In a more complicated case, patient would have high fever and hemolytic uremic syndrome. The organism can be identified from stool or blood specimens, using the conventional biochemical methods, PCR or PFGE. It was reported in a study that the prevalence of E. coli 0157H7 from beef samples purchased from retail stores in Malaysia was 35%. Of that percentage, 38.5% of the organism isolated were resistant to 4 different antibiotics. Even though thermal processing like boiling can inactivate this organism, but not the toxin it produced, which still imposes a public health threat. Therefore, further studies on how to effectively eliminate the organism and the toxin using methods that the public, such as food handlers, can apply should be done. Viral gastrointestinal infection is an inflammation of the stomach and intestines caused by viruses. The common symptom of viral gastroenteritis is usually watery diarrhea, that was caused from the impaired fluid absorption in the small bowel, due to cell damage from the virus infection. Other symptoms of the viral gastroenteritis is usually abdominal cramps, nausea or vomiting, and sometimes fever. The most common way to develop viral gastroenteritis is through contact with an infected person or by ingesting contaminated food or water. A healthy adult are likely to recover without complications. But for immunocompromised individuals like infants and older adults, viral gastroenteritis could cause complication that ranges from mild to deadly. There is no effective treatment for viral gastroenteritis other than rehydration and resting, so prevention is key. Prevention could be as easy as frequent hand washing to avoiding food and water that may be contaminated, and through administration of available vaccines. The most common causative agents are the rotavirus and the norovirus. They infect children in their early years and cause diarrheal illness, that may be severe enough to cause dehydration. In Malaysia, diarrheal disease is still one of the leading causes of childhood morbidity, and specifically rotavirus is responsible for 38% of hospitalization among children under 5 in a study population. The highest rate of rotavirus-associated diarrhea was among children aged 6 to 17 months, who accounted for 55.4% of all cases. Usually the stool specimen of the suspected patient are tested with the immunological techniques that targets the antigens, using methods like the enzyme immunoassay or in studying the ultrastructure of the virus using the electron microscopy. Since virus only contains RNA, the RT-PCR method is used to complement the RNA sequence, to form cDNA to get the genetic information, to investigate the suspected viral origins, and to relate them in epidemiological studies. The rotavirus is a sporadic, seasonal, and often the cause of severe gastroenteritis of infants and young children. Its main symptoms are vomiting, fever and watery diarrhea. In a more complicated case of rotaviral infection, the diarrhea could cause severe dehydration and ultimately death in children. The reservoir for rotavirus was said to only be in humans, and the virus is extremely resilient, as it could stay on surfaces for up to 10 days. The virus is highly communicable and transmissible, with as few as only 10 viral particles required to cause an infection. In an experimental condition, almost 50% of rotavirus remains viable on a contaminated hand for 60 minutes. Essentially, all children are infected by rotavirus in their first 2 to 3 years of life, which coincides with most outbreaks happening in child care settings or nursery. Other places of outbreak are the elderly home care, where most elderly individuals are immune-compromised. Healthy adults are usually asymptomatic to the infection. The methods of control are through the administration of oral vaccines, tightening the hygienic controls especially in child care settings, and preventing the exposure of infants and young children to people with gastroenteritis in the family. On the other hand, treatment was mostly focused on the rehydration therapy, as there is no antiviral treatment for rotavirus. In Malaysia, there was a suspected rotavirus infection outbreak in Perth 3 years ago which resulted in the deaths of 2 infants aged 2 and 10 months. In a 2004 study sponsored by the Ministry of Health Malaysia, it was shown that 1 in 37 children were treated as outpatients due to rotavirus, 1 in 61 hospitalized and 1 in 15,000 die from it. Most death from rotavirus is due to severe dehydration and organ failure. The World Health Organization figures show that approximately 400,000 to 700,000 deaths annually are related to rotavirus. The only intervention for this viral gastroenteritis, as recommended by the Malaysian Pediatric Association, is by incorporating vaccines, as the national immunization program for Malaysian children. Norovirus, or also known as Norwalk virus, have reported outbreaks in cruise ships, nurseries, hospitals and even catering events. As opposed to rotavirus, norovirus gastroenteritis could also occur in individuals of all ages, as the antibodies produced only reaches its peak at 11 to 16 years of age. The virus particles can cause an infection when as low as 10 to 100 norovirus particles are present. A high concentration of norovirus are usually found in the feces and vomit of an infected individual, which could be the source of the oral-fecal route transmission. Other than that, even the aerosolization of the viral particles present in vomits can contaminate surfaces, such as on the surface of a dish or in a food. So far there is no known drug or vaccine available for norovirus, therefore the treatment is mostly in managing the symptoms, such as oral rehydration therapy for vomiting and diarrhea, and analgesics for relief of headache. In a study conducted about the prevalence of norovirus in Malaysian food, raw vegetables like centella, bean sprouts and nialam were found to be a high-risk food, as vegetables are not subjected to heat treatment and norovirus was said to survive even at refrigeration temperature. Interventions such as using other sterilization method, to ensure the cleanliness of raw food could be implemented in the Malaysian food handling setting. I came across this interesting question from a patient to a doctor, asking about why he craves pizza in the midst of recovering from gastroenteritis. Accordingly, the bacteria that causes gastroenteritis kills, and reduces the probiotic lactobacillus in the intestine, which is responsible for the digestion of food. With the probiotic being reduced, the food becomes hard to digest. This sends signal to the patient's brain to ingest more lactic acid in order to produce more probiotic in the intestine, which makes the patient crave for cheese, or in his case, the cheese in the pizza, that is rich in lactic acid. The whole scenario tells us that probiotic could aid in the recovery of gastroenteritis, which is true from a meta-analysis study of treating acute gastroenteritis in children by using the probiotic strain of lactobacillus. From the study, it was concluded that the duration of diarrhea was effectively reduced with a high daily dose of probiotic. This discovery could potentially be an effective treatment that is applicable at home or in an everyday setting for children or even adults that is recovering from gastrointestinal infection.
About the infection about in the GI tract. Do you guys have any questions?
Maybe.
Yes.
May I know what is actually the most common mode of transmission for the bacterial infection in the GI tract?
Instead of stating all the mode of transmission for all these bacteria invading the GI tract, it's important to know that this bacteria's main goal is to control and manipulate the water regulation in the host of the GI tract.
I have one question. Why are most of the symptoms of the bacteria GI infections are usually diarrhea and abdominal cramps?
Because once the bacteria has to control and manipulate the host GI environment, the host will use the water regulation in the GI tract to expel the bacteria out, while the bacteria, on the other hand, will try to control the GI tract. Therefore, the diarrhea is generated from the synergy between the host reaction and the bacterial reaction in the GI tract. That's why. Any more questions?
Yeah. So, type of diagnostic can be used to investigate the outbreak of food poisoning in several restaurants that is speculated to come from the same source.
Okay, so if the outbreak is suspected to come from the same source, like polymerase chain reaction or the PCR, or the pulse field gel electrophoresis, or easily called as PFGE, can be used to get the bacteria's DNA information to determine if the bacteria come from from different restaurants are actually genetically related. Sorry.
Both rotavirus and norovirus are highly communicating now. But which viruses are more prevalent in older children to adults?
I would say the rotavirus is mostly contained in infants and younger children, or immunocompromised individuals like old people, because a normal healthy adult would have achieved the immunity. But for norovirus, on the other hand, the antibodies produced in the body would only reaches its peak about 11 to 16 years of age. So with that, infections could still occur in older individuals as long as they are not fully immune to norovirus. Yes.
Actually, for those people who have lactose intolerance,
Right.
and who have just had food poisoning, is there any other option for them to take the diarrhea food to replenish the intestine with probiotics bacteria?
Yeah, there is. There are options of non-lactose dairy products, such as almond meal and soy meal. Other than that, if you don't like meal, the commercial pharmacies do sell probiotic pills that contains lactobacillus that is essential in the intestines.
Okay.
Any more questions? All right, thank you.